Okay, today I will talk about Western blotting. This is a technique which you people practically you will need it, and theoretically it is there in your exams if you are in this field. So let's go to my slides on Western blot. Western blotting দেখা যাচ্ছে? Is it visible?
Yes, sir.
Okay, very good. Okay, so Western blotting is actually a laboratory method used to detect specific protein molecules from a mixture of proteins. This mixture can include all of the proteins associated with a particular tissue or cell types. They can be used to evaluate the size of a protein of interest and to measure the amount of protein expression. 1 second. You wasted. So basically, Western blot, it can be also used to evaluate the size of protein of interest and to measure the amount of protein expression. It's also called protein blotting. It's also known as immunoblotting. So essentially what you do, you have a cell extract. You have a cell extract and you want to check with that if that particular cell has gotten that particular protein expressed. So what you do for that is you crush the cell, you make an extract, load it on an SDS polyacrylamide page gel on in an electric field, and it separates. The protein separates in an electric field. Now it has separated on the gel, but now you need to detect it. You have to want to know your particular protein of interest, say insulin. So what do? You there it was a nice technique. You just transfer it onto a membrane. A membrane is nothing. It is just a nitrocellulose piece of paper. That membrane, you transfer it to that membrane through a technique known as Western blotting. You blot it in that, and then on that membrane you do some treatment and give antibody, and then the antibody will bind to your particular protein, and then you can see that particular protein. Also you load some markers, then you mark the markers on your membrane with a pencil, then you can see the size. So it's also known as protein blotting. You can evaluate the size of a particular protein, if that protein is expressed or not, how much it is expressed. This can be done with the help of Western blot. So it was developed by George Robert Stark and used antibodies to locate proteins. It is based on the principle of immunochromatography, where proteins are separated into polyacrylamide gel according to their molecular weight. So, in SDS page, what happens is that according to the in an electric field, in the last class, like I told, in an electric field, you can separate the proteins one by one according to the size. The smallest protein will be below, and the larger protein will be on the top. So these proteins are separated into on polyacrylamide gel according to their molecular weight, heavier on the top, lower on the bottom. And also in one particular well, you also load, what do you load? You load a marker. That is a mixture of protein whose sizes you know. That say 100 kilodalton, this is 100 kilodalton protein, 90 kilodalton protein, 80 kilodalton protein, 70 kilodalton, like the 10 kilodalton difference, you can have a marker. So in the neighboring well, corresponding well, you can know the size of the protein based on the size of the marker. So you know exactly how it is done. So the proteins are then transferred onto a nitrocellulose membrane. Basically, it's like a paper, thin paper. and are detected using specific primary antibody and a secondary enzyme labeled antibody. This we will explain, which in the end we will get a colored product. You can get a colored product, you can get a autoradiographic signal, you can get anything. So the primary antibody will bind, but since you can't detect the primary antibody, you have a secondary antibody which is against the constant region, the FC region of the primary antibody. And that antibody has got a particular, say, enzyme attached on its back. So the moment you give a colorless product, that enzyme will convert it into a colored product, and then you can actually see the band on the membrane, that colored thing on the membrane. The color indicate the presence of protein of interest. Is it clear so far? Yes, no?
Yes, sir.
Okay,
Yes, sir.
okay. So the procedure is, first you have to prepare the sample. You have to break open the cell, you have to grind it, you have to give HDS buffer, you have to heat the sample so that the proteins get denatured and HDS binds to it completely. Then you do the gel electrophoresis, then you do the blotting on the membrane. Then a thing does a procedure known as blocking. You use BSA or milk, 5% milk, to block it. That is on the membrane, all rough surfaces which are there, different shapes are there on the membrane. It's nothing in the world is completely flat. So what you do is you give a blocking, some protein or milk, it will block those particular shapes over there, so that the antibody will not bind to unspecific regions. Suppose a similar shape is there somewhere on the gel. Suddenly you see that the antibody will go and bind, and then you will see a color over there. So since that doesn't, so that that does not happen, you block it with milk, because milk will go and cover up all this region. Then you give primary antibody. Antibody will go and bind to your, in the gel, in a well of the gel, you will see that it will bind to that particular region. First primary antibody, then you wash the primary antibody off, then the secondary antibody which will bind to the primary antibody, and the secondary antibody has got enzyme linked to it on its back, and on that you add a colorless substrate, and that will get converted into a colored product. This is the basic thing. Let us see the details. So the first step is the sample preparation. That is, you isolate the protein from a sample. Usually proteins are purified from cell. Next, the protein concentration is determined. You determine the amount of, first you purify the protein from the cell extract. All the proteins you extract. There are ways of extracting proteins from the cell. So you extract the proteins, measure what is the concentration, so that you, in each well, you can load fixed amount of proteins. 10 nanomicrograms, so 10 micrograms. All the wells will have 10 micrograms. Next, the protein concentration is determined. Sample buffer is lamelli buffer, or which contains SDS and beta marcaptoethanol, not NOD. It's NOL. This is a, it smells like rotten egg, beta marcaptoethanol. एकदम बहुत बदबूदार चीज है, गंदा स्मेल आता है इसमें, लाइक रॉटन एग्स. और स्टमक अपसेट हुआ तो जो गैस निकलता है, सो डर्टी स्मेल, दैट समथिंग लाइक दैट, इट स्मेल्स लाइक दैट. So this is added to the protein suspension. Then it is heated to boiling, and this denatures the protein and allows SDS to bind to the protein. And this makes the protein linear, and so that SDS coats it completely. It is coated with the negative charge. So now the migration is not on the basis of the net charge of the protein, but on the basis of the size, because the everything more negative charge, the bigger the protein, less negative charge, the smaller the protein. Okay, so loading buffer, you heat it up. Before SDS, what happens? Plus, minus, all charges are there. So the migration in the electric current, it will be only on the basis of mass as well as on the charge. So after SDS, all the proteins are linearized and all it's covered by negative charge. And this is the reason that in this Eppendorf, they heat it like this. The second step is electrophoresis. So the it will there are some wells on this in this buffer, so you load the protein samples over here, and in one you load the marker, and you run it in an electric field. It will migrate in the electric current. The smaller molecules will migrate fast, and the larger molecules will be held back. It will be retarded. Next is the blotting. It is placed on a nitrocellulose membrane, pressure is given, and some electric current is given. And the proteins from the gel, it will migrate into the membrane. It is not 1-2 days. Nowadays, you use electroblotting, and that electroblotting takes 1 hour to 1.5 hours. Sufficient amount is transferred. But former times, they used it used to take 2 days to transfer. Just if you just place it on the top with buffer and paper towels and a large weight on the top. Now let's see, somebody is entering. Sound Sundar is entering. Okay. And electric current and the transfer to the protein on the membrane. So basically, it's like this. This is the gel, this white paper, this blue paper is the membrane. You add wet filter paper, wet blotting, and you sandwich it between two weights, minus and plus, and you press it and keep it and pass electric current. So gradually, what happens after you set this up? It is placed in this particular electroblotting, and the protein is transferred onto the membrane. Next is blocking. The membrane has the ability to bind to proteins. In this case, both target and antibodies are proteins, so there could be unwanted binding. So different shapes, so that you use milk or BSA, non-fat dry milk or BSA, bovine serum albumin, 3 to 5% milk. I normally prefer 10% milk. I used to use that. Blocking would be better. But in 10 minutes, 10% milk is much better. But if you use 5%, you have to block it for 1 hour. So the protein in the dilute solution attaches to the membrane in all. So how do you add the antibody? If you block it with milk, then in 10% in 5% milk you add your antibody, and then that milk containing antibody is poured in a box which contains your membrane, and it's allowed maybe overnight. You can keep it overnight, and within 1 hour I could see detection. It depends on how strong your antibody is. Now you treat it with antibody. The antibody is in milk. You can even then take this antibody and store the antibody back in the fridge. If you add to that what's that poison, that hydrogen cyanide, sodium cyanide, if you add a bit of sodium azide in that, sodium azide, now I remember that name, sodium azide, then you can keep it in 4 degrees, then the milk will not get destroyed. Or you have to, if you don't add sodium azide, then you have to freeze the milk. Every time you freeze it, you have to thaw the milk, and then you add it again. When your work is done, you pour the milk back in that container and freeze it back again. Sodium azide, you can keep it at 4 degrees, normal fridge. You don't have to keep it in the freezer. So this antibody, you can use it till it becomes weak. You can use it many times. So this antibody will bind to it is added, it will bind to your specific protein. See, on the membrane, it will bind to your specific protein. The protein are transferred over here. Now you add a secondary antibody. That secondary antibody which binds to an FC region. FC region is same for all antibodies. The FAB region, the variable region is different. So this FC region of the secondary antibody, it has got an enzyme attached, and the FAB region will bind to the primary antibody FC region. So primary antibody will bind to the protein, to the primary antibody will bind the secondary antibody against the FC region. It is same for all antibodies. So it will bind there, and this FC secondary antibody FC region has got an enzyme attached. It can be alkaline phosphatase or horseradish peroxidase. I have used both. Alkaline phosphatase converts a colorless substance into a brown color band. Horseradish peroxidase, you have to use a luminol reagent, and you have to use uh, a film, or keep a film on top, and that picture will develop. So on the basis of the size, you can see. But the easiest is alkaline phosphatase. For this, you need a photo machine where you have to develop a developer fixer and then water wash, and then the film will develop and come. Then you can see your bands. Detection, the substrate will be this protein, primary antibody against the primary antibody, secondary antibody. Here the enzyme is linked. The substrate will convert into product. You can see either a light which will be detected on the film, or you can see a colored product will be formed on the top of the band. So this is the procedure of Western blotting. Do you people have questions?
No, sir.
ওকে, তাহলে এইটা পড়ে ফেলো ভাই পুজোর মধ্যে, তাহলে পরে পুজোর পরে একটা অনলাइन क्लास নিয়ে নেব।
Okay, sir.
ओके, पूजा के बाद एक ऑनलाइन क्लास लेंगे, और
ओके सर।
10th के बाद 10th 11th के बाद ऑफलाइन क्लासेस चालू कर दूंगा फिर से।
ओके सर।
ओके ओके, बाय बाय, हैप्पी पूजास।
Thank you,
हैप्पी
sir.
पूजा, थैंक यू सर।
थैंक यू सर।
वेलकम।